I am honored to introduce our keynote speaker, Dr. Robert M. Califf, as the Commissioner of Food and Drugs. And as the top official of the FDA, he is committed to strengthening programs and policies that enable the agency to carry out its mission to protect and promote the public health. Previously, Dr. Califf served as Commissioner of Food and Drugs from February 2016 to January 2017. Dr. Califf also served as the FDA's Deputy Commissioner for Medical Products and Tobacco from February 2015 until his first appointment as Commissioner in February 2016. We are truly honored to have Dr. Califf here in person. We know how busy his schedule is, so please let us welcome Dr. Califf.
Thanks so much, Brenda. I know these meetings take a lot of work, and it's much appreciated. If I look a little tired, I've spent the last 4 days chasing my 3- and 4-year-old grandchildren while their parents were taking a vacation at 11,000 feet in Breckenridge, where they live. So just returned last night. It's amazing the energy that 3- and 4-year-old children have. And, so I am delighted to join you to help kick off today's workshop. This marks the 7th year we've held this important event, focusing on the transformative impact on generic development, regulation, and approval as a result of the FDA's generic drug user fee amendments, science and research programs. These meetings take a lot of work, and they summarize a lot of information. It's a great chance for me as I prepare for this to summarize from the point of view of the FDA as a whole where we are. And it is important, this is the first year that's included both in-person and virtual attendees after 4 years of virtual-only workshops. That's an exciting development, and I want to thank the Office of Generic Drugs and Office of Pharmaceutical Quality in collaboration with the Office of Small Business and Industry Assistance for facilitating this and once again organizing today's workshop. I also want to recognize the developers and other stakeholders who are participating here today, as well as the industry support of the generic drug development program. The FDA offers many different types of public workshops and meetings, but today's has a particularly important emphasis. At the most basic level, this meeting provides a valuable opportunity for attendees to engage with FDA scientists on topics including guidance development, advanced analytics, and physiologically based pharmacokinetic modeling for generic drug development and equivalence characterization. This is an approach we pursue across the agency in our effort to provide meaningful opportunities for FDA scientists and industry to share knowledge through personal contact and in-depth discussion. What makes this session even more special is the focus captured in the second part of the program title, Translating Science to Approval. These few words highlight an important aspect of the FDA that's often overlooked or misunderstood, and that's the role we play in spurring and supporting innovation. A critical part of our work to advance the health and safety of consumers and patients involves our role in driving scientific innovation that will benefit the public. We're constantly working to develop and adapt regulatory policies in response to and in anticipation of scientific opportunities and changes so they can be harnessed for the improvement of health and medical care. Few areas have as direct a connection to the public in this regard as generic products, both in the personal benefit that generic products provide to patients and the broader advantages they bring through lower costs and increased accessibility as a result of greater competition. I certainly can speak from personal experience to the benefits of various medications I've taken over the years that are available to me in generic form. As a cardiologist, I'm keenly aware of the importance of controlling my blood pressure and lipid levels as I age. This is completely accomplished with inexpensive generic drugs, and I'm sure many of you have had similar experiences. It's no coincidence that this meeting is being held today, September 24th, a date that marks the 40th anniversary of the Hatch-Waxman amendments. That law has had a remarkable impact on the U.S. healthcare system by creating the modern generic drug application pathway and strengthening the generic drug industry, which in turn has provided the public with greater opportunities to acquire less expensive treatments. It's truly a paradigm shift. Consider that when the amendments were enacted in 1984, Generic drugs accounted for just 19% of all prescription drug purchases in the United States. Today, generics account for more than 90% of prescriptions filled in the U.S., and as the pipeline from the innovative sector ages, the availability of generics continues to rise. The economic effect is also significant. According to the Association for Accessible Medicines, in 2022, the most recent year for which statistics are available, generic drugs saved the U.S. healthcare system an estimated amount of almost $400 billion. I feel compelled to also point out that a high-quality, low-cost, and reliable generic drug supply is essential to the public health of the more than 7 billion people who live outside the United States. Your success has implications far beyond the needs of the U.S. and other high-income countries. But as in any field, and nowhere more so than in science, changes come fast and frequent, and those changes require continued adaptation. When the Hatch-Waxman amendments created the framework for a generic drug review, most drugs were simple small molecules. These products generally involve straightforward and relatively simple reproducible manufacturing processes. They were easier to characterize and evaluate through traditional methods, including human bioequivalence studies. Today, however, many drugs have more complex formulations, active ingredients, and delivery systems. In other instances, a complex drug may be the one that acts locally on the tissue rather than through the concentration in the blood. What all this can mean is that these products can be more difficult to replicate, develop, and manufacture. And this is where the FDA plays a particularly important role. As the pharmaceutical science advances and the challenges posed by the increasing complexity of formulations and delivery systems grows, the FDA works to provide generic drug developers a clear regulatory process with streamlined, predictable, and science-based information that help reduce the time, cost, and uncertainty in developing these products. FDA scientists work collaboratively with partners in academia, the commercial research infrastructure, and industry to communicate early and develop innovative tools to overcome some of these challenges. The centerpiece of this process, as well as of this meeting, is the 2012 Generic Drug User Fee Act, or GDUFA, and its subsequent reauthorizations. GDUFA introduced critical timelines for application assessment. Most significantly, though, for our discussion here, it established a GDUFA Science and Research Program, which, thanks to the dedicated resources and structured framework for innovation, continues to help propel the industry forward, driving progress in science and technology to enhance generic drug development and approval. The program has consistently fostered early engagement between the FDA and generic drug industry, supported collaboration with generic industry representatives to determine the priority initiatives, facilitated continued engagement with prospective ANDA applicants through pre-ANDA meetings, and provided opportunities for better informed engagement following ANDA submission in meetings between FDA and ANDA applicants to discuss scientific matters. Here I'd like to just say I would encourage all of you to talk this up, because when I'm dealing in the public sphere, very few people actually understand how much work goes into the pre-application system and the importance of the communication that goes on. It's a huge part of the work of the FDA that I think is not well appreciated. At its core, GDUFA research aims to make generic drug development and assessment more efficient while maintaining a high degree of reliability. These efficiencies can make it more feasible for manufacturers to develop generic drugs, which can reduce the risk of drug shortages and facilitates competition that generally reduces the cost of medicines. This enhances patient access to treatment by helping make these products more available, allowing patients in the U.S. to get the medicines they need. Before I return to the evolving landscape of generic drugs, I think it's important to address the occasional criticism and generalization, or perhaps overgeneralization, that we often hear from some who say, quote, generics are just copies, unquote. On one level, of course, that's true. Generics include the same active ingredient formula as a brand name drug. However, the development process involves far more than simply copying ingredients, as you might do in a recipe. It involves intricate scientific work, and by the same token, assessment across various disciplines is required to evaluate generic drug applications to ensure that generic medicines are safe and effective before they can be approved. Both the development and the assessment will serve society best if we collaborate on the principles of the involved science in manufacturing while maintaining appropriate boundaries in the evaluation process. To use a much simpler example, consider what it would be like to replicate your grandmother's famous and probably top secret chocolate chip cookie recipe or the secret formula for Coca-Cola. You couldn't do it by just looking at the cookies or the soda. With a generic drug, however, you're not just trying to replicate taste, you must demonstrate bioequivalence. That's a tough standard involving a complicated process. And it's only grown as the products increasingly involve some of the most advanced pharmaceuticals in the therapeutic armamentarium. They may involve novel delivery systems with products that include precision devices, novel active ingredients such as oligonucleotides, and unique routes of delivery that are relevant to specific diseases or provide more rapid onset of efficacy. Moreover, the process involves more than just scientific review. We also have investigators who conduct facility inspections throughout the world. We're in the middle right now of the largest reorganization in the history of the FDA, largely involving the group that you used to know as ORA that will be known as OII as of October 1st as the reorganization comes into place. And our patent exclusivity team plays an essential role in determining if we can act on applications. Furthermore, disciplined project managers and regulatory project managers help pull all these pieces together. All of this underscores the complexity and enormity of this undertaking as this field of regulatory science continues to evolve. The FDA works to integrate technological advancements and innovative research methodologies to streamline approval processes. Innovative tools enhance the understanding of complex drug interactions and mechanisms, aiding in the development of generics for more accurate, intricate formulations. Robust approaches, including analytical techniques and bioequivalence studies, enable accurate assessment of generic drug quality and equivalence to ensure that generic drug products are safe, effective, and high quality, just like their brand name counterparts. Let me offer one example of a recently approved generic product that illustrates the meaning of FDA-supported scientific innovation and what can come out of years of GDUFA-supported research. In July 2024, FDA approved the first generic for bupivacaine liposome injectable suspension, a locally acting product for pain management. The treatment uses multivesicular liposomes as the drug delivery system. By any definition, it's a complex product. It was the FDA's research to better understand formulation characteristics and drug release mechanisms, which was presented at this workshop in 2018, that enabled the development of a product-specific guidance that lays out clear expectations on an in vivo PK study in healthy subjects, along with comparative characterization studies to enable generic development of this complex product. And last October, the Generic Drug Program launched a pilot that offers new and early meeting opportunities to prospective generic drug applicants who are using model integrated evidence approaches for bioequivalence establishment in their ANDAs. MIE approaches combine data from in vivo or in vitro studies with scientifically sound mechanistic models to provide more efficient ways of demonstrating bioequivalence in some of the most complex situations facing the generic drug program. I also want to briefly mention how the FDA's research and collaboration extends to other international regulatory authorities in our work to create consistent, scientifically driven international standards. The recent endorsement by the International Council of Harmonization of the first harmonized guidance for generic drugs titled M13A, Bioequivalence for Immediate Release Solid Oral Dosage Forms, is one such example of successful collaboration. This guidance, which represents efforts and harmonization of 22 global organizations, describes the scientific and technical aspects to support bioequivalence assessments for orally administered immediate release drugs. You'll hear updates on all these programs during the workshop, and I hope many of you will take advantage of FDA's many opportunities to engage during the drug development and approval processes. Finally, I want to mention the logistical challenges involving supply chains that we're working to address. For instance, one way we're trying to strengthen our medication supply chain and patient access is by encouraging and facilitating the adoption of advanced manufacturing processes. Ensuring that drug manufacturers will have access to advanced manufacturing technologies is vitally important because advanced manufacturing requires a skilled workforce and can help domestic companies operate with lower costs and fewer quality defects in smaller facilities, improving the global competitiveness of U.S. manufacturing. Our Center for Drug Evaluation and Research, or CDER, has developed an emerging technology program known as the Framework for Regulatory Advanced Manufacturing Evaluation Initiative, or FRAME. Its goal is to support implementation of advanced manufacturing technologies and build a robust extramural and intramural advanced manufacturing research program. The program has a purpose to engage with companies that are pursuing innovative approaches to pharmaceutical product design and manufacturing early in the development process. The emerging technology team can help identify and discuss scientific and regulatory challenges prior to a regulatory submission, which can streamline the application submission process. The ETT has facilitated the approval of drugs manufactured using continuous manufacturing for a range of product types, including solid oral dosage forms and biotechnology products. As I hope this brief summary makes clear, there's a lot happening in this area, and we're eager to work with you. If you thought it was simple, just the terms that have been used in this address are really quite remarkable. If we look back 20 years ago, couldn't have been anticipated. This workshop is a key component of our stewardship of the GDUFA program and an example of our dedication to open communications with the generic drug industry and the broader public on this important research. By sharing cutting-edge research, methodologies, and tools, such as the new equivalence approaches, analytical techniques, and PBPK modeling, we seek to advance the development of generic drugs and contribute to the availability and access to safe, effective, and high-quality medicines. Thanks again for your engagement. I know there's a big audience out there who really are more interested in hearing from the scientific experts than the pontification of the commissioner, but I appreciate the chance to summarize a lot of great work done by our team and the great hopes for you all as you make better and better drugs more accessible to the public. Thanks again and have a great meeting.
Thank you very much, Dr. Califf. It was indeed a pleasure for you to make time on your schedule for us. So thank you so much. Okay, so let's enter into the world of science and complex generic drugs. And what I'll do, I'll introduce each session, all the speakers at once, and then each speaker will come up in turn as they present. So let's start with session 1, our scientific and regulatory considerations for in vitro release test for complex drug products. First, we will have Dr. Abu Mustafa, who is a pharmacologist with the Office of Bioequivalence within the Office of Generic Drugs. Then we'll have Dr. Thilak Mudalij, who is a research chemist from Arkansas Human and Animal Food Laboratory, still within the Office of Regulatory Affairs. And for the third speaker for this session, we'll have Dr. Dankai Zhu, who is a visiting associate from the Office of Pharmaceutical Quality Research within the Office of Pharmaceutical Quality. So let's first start with Dr. Mustafa.
Hello, good morning, everyone. Thank you, Brenda, for your kind introduction. So I am AGM Mustafa. I am a staff fellow in the Office of Bioequivalence. Welcome to my presentation. In this presentation, I will talk about in vitro release test, IVRT, for in situ gel or depot forming drug products. So what do we want to learn from these presentations? So in this presentation, I will talk about some key challenges in IVRT method development for in situ gel or depot-forming drug products. I will provide some key considerations in IVRT method development and validation for this type of drug products. I will also provide an overview of the important submission contents related to IVRT study in ANDA. So let us first understand what are these in situ gel or depot-forming drug products. So these are, in simple terms, these are specialized formulations which after administration forms a gel or depot at the administration site, and that drug releases from that depot over an extended period of time. So for this drug product, the in situ form depots dictates the drug release kinetics. So far, FDA has approved 15 new drug applications and 4 abbreviated new drug applications for this type of drug product. You can see that there are relatively fewer approved generics for this type of drug products. There are multiple reasons, including challenges that are associated with bioequivalence study. This slide contains a summary of the bioequivalence study recommendation provided in the product-specific guidances of in situ gel or depot-forming drug products. As you can see that for most of these drug products, PSGs recommend in vivo BE studies. However, there are drug products for which PSGs recommend in vitro BE approach also. And in many cases, for in vitro BE approach, IVRT study is a key component. Here we have 4 examples of in situ forming drug products for which PSGs currently recommend IVRT study. Okay, now what is the purpose of IVRT study here? So IVRT study here is the component of in vitro BE approach. The other 2 components are Q1, Q2, qualitative and quantitative sameness, and Q3, which is comparable physicochemical characteristics. In case of in vitro BE approach, the bioequivalence is determined based on the totality of evidence. And here IVRT is supposed to determine or ensure that the rate and extent of drug release, which can be affected by factors such as drug substance, formulations, and manufacturing process, the comparability, make sure that the test and reference products have comparable test and rate and extent of drug release. And by ensuring that, IVRT study mitigate a potentially major bioequivalence failure mode. Now, why is it challenging to develop an adequate IVRT method for this type of drug product? First of all, there is no compendial method available for this type of drug product to follow, and the product-specific guidances do not provide any specific recommendation about the study conditions. Also, there are challenges associated with demonstration of discriminatory ability of the proposed IVRT method. However, for this drug product, the most specific challenge is optimization of the gel or depot-inducing conditions. Now, how to address these challenges? So the key to address this challenge is to understand the mechanism of depot formation and drug release for these drug products. The process of depot formation and drug release can vary from product to product. For example, there are some in situ forming drug products that are self-aggregating peptide drugs, and also there are some polymer-based formulations. In the subsequent 2 slides, I will provide illustration of the process of depot formation and drug release for these 2 types of drug products. So in case of self-aggregating peptide drugs, after administration, these peptide drugs are usually administered as solution form, and after administration, the peptide drugs undergo a self-aggregation process. And this self-aggregation process of peptides forms a dense fibrillar network, which acts as a depot. And from this depot, the peptide drug diffuses out into surrounding tissues. So for this type of drug product, the microstructure of this fibrillar network dictates the drug release kinetics. In case of polymer-based formulations, these formulations usually consist of drug substance, polymer, and organic solvent. After administration, the organic solvent diffuses out and the body fluid diffuses in. And this process causes precipitation of the polymer with entrapped drug substance. And the precipitated polymer drug acts as a depot from which the drug releases over an extended period of time by various mechanisms such as initial burst release, which is followed by diffusion facilitated release and degradation facilitated release. And for these drug products, the characteristics of the polymer dictates the release kinetics for this type of drug product. So we have discussed that for in situ gel or depot forming drug products, the in situ form depots dictates the release kinetics. And that's why, unlike the conventional IVRT method, in case of in situ gel or depot forming drug products, the IVRT method requires a gel or depot formation step. To further elaborate on this unique IVRT method parameter, which is gel or depot formation. So in case of conventional IVRT, the drug product samples are loaded as it is onto the IVRT apparatus. However, in case of in situ gel or depot forming drug products, the drug product samples to be used in the IVRT should be in a adequately formed gel or depot state. If IVRT is conducted with samples that are not in gel or depot form or not in adequately formed gel or depot, then the observed release profile will not reflect the true or actual release characteristics of the drug product. Instead, we may observe some artifacts or misleading release profiles. And this will eventually lead to incorrect assumptions on the comparability of the rate and extent of drug release between the test and reference product. So what are the key parameters for IVRT method development for this type of drug product? The parameters are sample preparation, selection of apparatus, sample loading, release medium selections, flow or stirring rate, temperature, sampling time, and specifically for this drug product, gel or depot induction. For each of these IVRT method parameter, one should take a step-by-step systematic approach for selection of the most optimal conditions. And the expectation is there should be adequate justification with supporting data for the selected conditions. And the overall objective of IVRT method development for this drug product is to achieve adequately sustained release profile and at the same time complete release within a reasonable time frame. So what are the key considerations for IVRT method development? What are the key considerations for gel or depot induction for this type of drug product? First of all, one should take an exploratory approach to investigate the factors that can affect the gel or depot formation process. Since it is not practically feasible to exactly mimic the in vivo condition, one should take an exploratory approach, explore various inducing conditions, and including the physiologically relevant conditions. And depending on the drug products, some of these factors are sample amount, the inducing media, sample to media ratio, incubation temperature, and time. And after the trial, after the induction, one should assess the consistency and reproducibility of the gel or depot that are formed using the given conditions. Next, I will talk about the key parameters for IVRT method validation. The two important parameters for IVRT method validation are robustness and discriminatory ability. In case of robustness, we want to see that slight variation in IVRT method conditions do not have significant impact on the results. And there should have pre-specified criteria for robustness. In case of discriminatory ability, we want to see that the proposed IVRT method can differentiate intentionally designed non-bioequivalent experimental test formulations. And during discriminatory ability evaluation, one should consider the critical formulation and manufacturing process attributes that can affect the depot formation and drug release. Now, what are the key IVRT study-related submission contents that we expected to see in an ANDA? The submission contents should cover these key elements: IVRT method development, IVRT method validation, analytical method validation, and pivotal IVRT study. And some typical contents are standard operating procedures or protocols that are effective at the time of a study, the detailed study reports, sufficient experimental details that will enable the assessor to reconstruct how the study was conducted, and all the data and calculations, which will enable us to assess the accuracy of the results. From agency's point of view, all these informations are critical for the assessment process. And in many instances, it is found that the deficiencies are mainly due to incomplete submission, incomplete information in the submission, which eventually delay the approval process. For awareness purposes, here we have summarized some typical IVRT study-related deficiencies that are found in an ANDA for in situ gel or depot-forming drug products. So we have found that in some cases, though gel or depot formation is needed, but the proposed IVRT method, like a depot or gel formation step, and it is not clear from the submission that the samples, the IVRT samples, are in adequately formed gel or depot. In some cases, it is found that a random condition is selected for gel or depot induction without conducting thorough exploration of different conditions, including physiologically relevant conditions. And there are some instances we found that adequate justification is supporting data were not provided for the selected conditions, or the justifications were inadequate or incomplete. We have also found examples where the gel formation was inconsistent and irreproducible, which potentially contributed to the observed high variability in the release profiles. Another typical deficiency we found is that in case of discriminatory ability evaluation of the proposed IVRT method, the critical attributes are not considered. So here are some key takeaways from this presentation. First of all, the gel or depot formation is a critical step in IVRT method development for in situ gel or depot forming drug products. It is advisable to conduct a thorough exploration of different inducing conditions, including physiologically relevant conditions. And one should aim to achieve a well-formed, consistent, and reproducible gel or depot to ensure reliable and representative in vitro release profile. For optimization of IVRT method parameters, it should be based on the understanding of the depot formation and drug release mechanism. And during discriminatory ability evaluation of the proposed IVRT method, one should consider the critical attributes which can be related to formulation and manufacturing process that can affect the gel formation and drug release. We have two challenge questions here to assess if we have achieved the learning objective of this presentation. So the first challenge question is, which of the following statements about IVRT So I'll give you some time to go through the options. And the answer is option A and C. So in case of gel or depot forming, understanding the gel or depot formation and drug release mechanism is important for IVRT method development. And in case of in situ gel or depot forming drug products, additional consideration should be given to the gel or depot forming step. Challenge question 2 is, which of the following statements about IVRT for in situ gel or depot forming drug products are false? Select all that apply. And the options are and the answer is option A and E. So in case of IVRT method, it does not necessarily need to exactly mimic the in vivo release profile. And IVRT method validation is distinct from analytical method validation, and it is expected that the submission contents should cover both aspects. So I'd like to thank Office of Bioequivalence for giving me this opportunity to talk about this topic. And my special thanks to Hisan, Utpal, and Bing from Office of Bioequivalence who helped me with this presentation and guide me with this presentation. And last but not the least, the assessment teams from Office of Bioequivalence who reviewed these complex drug products. And the basis of this presentation is those assessments. Thank you so much for your attention. Next, I would like to invite Tilok Mudalis for his presentation.
Thank you very much. Today I will talk on Nano-Size compressed product in vitro release testing. I'm a research chemist from Office of Regulatory Affairs. So I will start my presentation with introduction to drug product containing nanomaterials, liposome and liposome drug product, in vitro release testing of compressed product containing nanomaterials and challenges. Finally, I will discuss innovative method we developed in our lab. So what is complex product? Drugs that are characterized by complex active ingredient, formulations, or route of delivery. What is drug product containing nanomaterials? Submicron size particles with one or more therapeutic agents that are dispersed, adsorbed, or covalently bound in encapsulated vesicles, capsules, or polymer matrices. Drug-loaded liposomes, polymer nanoparticles, or protein nanoparticles are prime examples for those products. So what is liposome? Liposome is microvesicle composed of lipid bilayer. It has a central aqueous cavity. We can use liposomes to deliver drugs. If active pharmaceutical ingredient is hydrophilic and water soluble, we can load into the central cavity, aqueous cavity. If active pharmaceutical ingredient is hydrophilic, we can load into the lipid membrane. There are many different kinds of liposomes based on the structure of lipid membrane arrangement. They can have a single layer of single lipid bilayer is called unilamellar liposomes. Multiple concentric lipid bilayer is called multilamellar liposomes. And they can have a multiple compartment in single liposomes separated by lipid membrane. It's called multivesicular liposome. When liposome is loaded with active pharmaceutical ingredient, it is called liposome drug product. So how we do in vitro release testing for liposome drug product? So we set the condition for drug release in drug release media. We can control pH, temperature, and media composition and other parameters. Then we add the liposome formulation to the media. So it starts releasing active pharmaceutical ingredient and to the outside of the liposome. Then we collect the sample each time point. Once we collect the sample, we need to separate them immediately. Collecting sample does not mean it stop releasing. Even we taking the sample out, it keep releasing. So we need to separate them immediately and then quantitate the released drug to make a drug release profile as shown in last diagram. But this is challenging process because both nanoparticles and active pharmaceutical ingredients are same size. So it is difficult to separate by conventional technique. And also these liposomes are delicate structure. If you use harsh conditions to separate them, they can break open. So when we develop a drug release testing method, we need to consider all these parameters. There are mainly two different ways to do drug release testing for liposomal formulation. One is as shown in previous slide. We can take sample and then separate externally, either filtration or centrifugation. But both filtration and centrifugation is challenging because active pharmaceutical ingredient can bind to the membrane or liposome can get disintegrated. Sometimes difficult to centrifuge because lipids and media has same density, so they don't get sedimented with centrifugation quick enough. Other method is membrane diffusion method. We can take a dialysis sac and then load liposome inside the dialysis sac and place dialysis sac in the drug release medium. And drug release out from the liposome released to the surrounding medium, and then we can take samples from surrounding medium. We can do in the container, or we can do this on flow-through system like a USB type 4 dissolution system. But this is also challenging because active pharmaceutical ingredient can bind to the membrane in this case. Also, it's a non-sync condition most of the time. That the drug release inside the dialysis cassette that does not come out immediately. There's always concentration gradient. With considering all the above challenges, we set up the condition to develop new drug release testing method. We need to eliminate manual separation, avoid artifact associated with disruption of nanoparticles, and we need to do it automated way, and real-time sampling and analysis, because even after we take the sample, active pharmaceutical ingredient keep releasing, we cannot store the samples. So first example I'm showing is application of capillary electrophoresis to do automated drug release testing. Capillary electrophoresis is a very gentle separation technique. It uses buffer-filled glass capillary, and once we inject sample from one side, we can apply electric field and we can separate released drug from liposome bound drug. And then we can use optical detection, like the optical absorbance or fluorescence. Even we can connect to the other detectors and then measure the released drug. So this is automated method. And this buffer we use in this method for separation is compatible with liposomes. And this method need very small volume of sample. It is a fraction of 1 microliter. Few hundred picoliters. And also we can do simultaneously take the sample out, separate and quantitate in automated manner. So this is the example of use of capillary electrophoresis for drug release profiling of liposomal doxorubicin. In this case, the doxorubicin is loaded inside the liposome. Once we set the drug release condition, it starts releasing active pharmaceutical ingredient. And then we can set up the instrument to take the sample directly from the drug release vial and then separate from bound drug to pre-drug as two separate peak. And then we can integrate the peak at the end of the experiment and make a drug release profile. Here I'm showing a drug release profile of 5 FDA-approved doxorubicin liposomal formulations, and more details available in a recent publication. We can use same method for drug release profiling of visudine. Visudine is a liposomal formulation of vertepropine. Vertepropine is water-insoluble compound. So once we inject this formulation to our blood circulation, it releases as a human serum albumin complex, as it is water-insoluble. In this case, we use human serum albumin to assist the active pharmaceutical release as a complex, and we can separate the released drug from liposomal bound drug. So drug release condition for this drug is we need to get 90% of active pharmaceutical ingredient released in 10 minutes. And we changed the vertepropine to human serum albumin ratio and did the experimentation. And even at 1:1 ratio, we can get out 90% of the active pharmaceutical ingredient. In the human blood, we have about 35 gram to 50 gram of human serum albumin, and it has much more human serum albumin than it wanted to release the drug. So this experiment was very successful, and more detail available in recent publication. So next, I'm going to show a different way of doing drug release testing. In this case, we use electroanalysis. So we insert 3 electrode to drug release container or drug release vial, and then we can in situ measure the released drug without taking any sample. So here in this case, we use the redox reaction of doxorubicin, whatever coming outside the liposome, and then redox current is proportional to the concentration of doxorubicin. So in that way, we can measure the drug release in situ without taking any sample, and then we can generate the drug release profile as shown in this figure, and more detail available in our recent publication. We also develop other method. One of them is a chromatographic method. We modify HPLC system to develop a drug release profiling method. Here we use a monolithic silica column coated with polyvinyl pyrrolidine. And this column has 1 micron through pore, so those pores go all the way along the column. And when you inject the liposome to this column, it let liposome pass through the pores and then release active pharmaceutical ingredient bind to polyvinyl pyrrolidine, and then we can elute them later. So we can separate liposome from released drugs and generate the drug release profile. This is the drug release profile of FDA-approved 5 liposomal formulations. And again, more information available in recent publication. So each of these methods has advantages and limitations. So we have to carefully select the method when we use it. Capillary electrophoresis use very small sample volume, so that's an advantage. And also it's a delicate separation technique because separation happens in aqueous phase. But separation is done by applying electric field, and only charged active pharmaceutical ingredient can be separated by this method. That's the limitation. Electroanalysis is another way to do the in vitro drug release testing. So this is advantage, no separation, no sampling. We can set up the instrument and it measure the released drug automated way. But this method only work with electroactive APIs. So these compounds should have a redox or oxidation reaction. The other method I discussed is a chromatography-based method. This method highly accurate. It take exact sample volume. If they take 1 microliter, it's exactly take 1 microliter, and it analyze the sample. Also, we can use the multiple detectors. Some of the active pharmaceutical ingredients are not optical detection cannot be used with some of the APIs. So we can even set up to the mass specs for quantitation. But this method need a specialized column compatible with the formulation. If we use a column which is not compatible, that can break open the liposome and then give erroneous results. Finally, I would like to summarize my presentation. So we developed an innovative method for drug release profiling with a real-time sampling and analysis for liposomal product. Also, active pharmaceutical ingredient specific physical and chemical properties should be considered for selecting in vitro release testing. So we have to look at as I shown in these 2 points, we have to look at the compatibility of the liposome formulation with the method. And also detection method has to be compatible with the active pharmaceutical ingredient. And these methods can be utilized for other liposomal formulation with careful selecting the compatible method. Finally, I would like to acknowledge my co-workers at Arkansas Laboratory. They are the one who did all these experiments, including my lab director listed at the end. Also, I would like to thank ORS, OGD, CEDAR. This work done as a collaboration between myself and Dr. Wenley. And also I received technical support and financial support from CEDAR, OGD, ORS. Also, I would like to thank Office of Regulatory Affairs, Office of Regulatory Science. They provide financial support and technical advice for my work. Thank you for listening my presentation. Next speaker is Dr. Dong Kai Shu. I'm inviting him to give his presentation.
Hi. Good morning, everyone. My name is Dong Kai Zhu. I'm a researcher from Office of Pharmaceutical Quality Research, OPQ CEDARS. Today, I'm going to share with you some results about how we develop in vitro release testing for complex drug product. Okay, so everyone deserves like confidence in their next dose of medicine. So pharmaceutical qualities assure the availability, safety, and efficacy of every dose. So this is the overview. So my presentation will have two sections. In the first section, I will talk about why we need better in vitro release testing methods for complex drug products, including challenges and needs. And in the second section, I will talk about our internal approach to solve these problems. In this section, I will use adaptive perfusion as examples, and then followed by a case study and some other potential uses. So like in vitro release testing, also known as IVRT, has been designed for measure the in vitro release rate and extent of the drug release. And IVRT has been widely used for product development, quality control, and bioequivalence studies. So the pictures here are common IVRT methods. So the methods that with blue name tags are compendial methods, like for example, like USB apparatus 127, and the methods with red name tags are the non-compendial methods. So both of them can be used to measure the in vitro release behavior for complex drug product. So the scope of this talk is focused on the challenge of the developing IVRT method for complex drug product like ophthalmic emulsions, protein bonded complex, and also the long-acting injection product and nanomaterials. So you might wondering what a good IVRT method, what kind of IVRT method is a good considered as good. So a good IVRT method needs to be reproducible, which includes being precise and robust. And also it needs to be discriminatory, which including like sensitivities and selectivity. So the goal of IVRT, like I just mentioned, is to measure the in vitro release. The goal of IVRT is to measure the in vitro rate and extent of the drug release by separating the free drug from its other carriers. And the common ways to use this is to membranous diffusion. So which is, and this method can be applied to many type of drug product like Dr. Robinson liposome injection and paclitaxel protein bonding injectable suspension, nano emulsions, and nano suspensions. So however, the membranous diffusion does have the limitations because it's covered by the concentration gradient from the high concentration to the low concentration side. And also the diffusion membrane is slow. So usually it takes about 2 to 3 hours for 90% of the drug come across from the membranes. So compared to the drug release, which usually take about within a couple of minutes. So the membrane diffusion method maybe become a rate limiting step and cannot capture the entire process. For example, the release process you saw may not be the truly release process. For example, some of the complex formulations that exhibit multi-phase release, like birth release and the earlier stage, the membrane diffusion method may not be captured these features. So now I would like to use the ophthalmic emulsions as examples to further discuss these questions. Ophthalmic emulsions, because it's considered as one of the type of the complex drug product, and it can be challenging because it may contain active ingredients like cyclophorin. And also ophthalmic emulsions have complex dosage form and may exhibit multi-phases release characteristic. And also ophthalmic emulsions, it has contained the complex route of delivery, which is through the eyes. So all those features that make ophthalmic emulsions is a good example for complex drug products. So here is the result that we did for the that we use a reverse dialysis as the IVRT method for measure the in vitro release behavior for diphosphorin ophthalmic emulsions. The result on the red shows that in these studies we test like 2 different formulations with different globule size distributions. On the right side, you can see there's no discrimination was observed between this formulation in terms of the release profile. And also there's no multiphysics behavior was observed. And because of this too slow release rate, product degradation was detected. So this kind of highlights the limitations of the traditional dialysis-based IVRT method. So the challenge with the current IVRT method can be divided into two sections, which is reproducible and discriminatory. So from the previous example, we saw the reproducible is well presented in the result by seeing this really tiny error bar in the figures. So how can we improve this IVRTs? So there might be different ways to do that, to tackle this question. And for example, like by developing any other type of in vitro release testing system. So here is an example that I'm going to talk about is that what we did in our lab. This IVRT method named as adaptive perfusion system. So adaptive perfusion system is utilizing the tangential free filtration principle to study the drug release. So compared with the normal filtration process, there will be the feed flow for this method will be parallel to the membrane filters. So there will be really less accumulations on the membrane surface. So anything that accumulates on the surface will be swiped out by the feed flows. Also, anything that's smaller than the pore size of the filter will be forced to migrate through the pore size and to achieve the separations. So there's two vessels are used in this method. One is named as retentive vessel. One is named as permeate vessel. So the retentive vessel is used to study how much drug was remaining in the system, and the permeate vessel is used to study how much drug was released in the system. Because the entire system was recording, so we are easy to calculate the rate and extent of the drug release. So this is a schematic of the adaptive perfusion system. So if you see in the middle, the blue vessel is the retentive vessel, and where you inject samples here and mixing with the dissolution medias. And the gray vessel on the left is a fresh pump reservoir. It will continue to pump fresh media to the retentive vessel to maintain the constant volume in the retentive vessel, which is very critical to calculate the drug removal rate. And the green reservoir on the right is a permeate reservoir, which is to collect the drug release the release drug. And let me explain how this system works. So at the beginning, so you inject sample into the retentive reservoir, which is like the red line here, and the mixture, and then pump through the TFI filters and go back to the retentive reservoir, followed by the blue line. And the back pressure controlling valve here will create a transmembrane pressure that is perpendicular to the feed flow inside the TFI filters. And this pressure will serve as a driven force that forces those free drugs that's smaller than the pore size of the TFI filter to go through the permeate pathway and collect by the permeate vessels. And because of the continuous pumping from the fresh medium, the liquid volume in the retentive reservoir will stay constant. That is really critical. So for us to calculate the drug release rate. And here is a case studies. So we are using adaptive perfusion as an IVRT method to study the diaphragmatic ophthalmic emotions, and we intentionally create 3 formulations with different globule size distributions. And in this study, we also include the pure drug solution and drug-loaded micelle solutions. And on the left figure, you can see the rate and extent of the drug has been removed from the retentive reservoir. And on the right figure, you can see the in vitro release behavior for these 5 formulations. So what we can see from the left figure is that really like 80% of drug has been released from the solution in less than 10 minutes. And similarly, what we saw from the red figures is that almost 90% of drug has been released to the retented to the permeate vessel within 30 minutes. So both of the figures shows that adaptive perfusion can achieve the fast release, and the figure on the permeate side too shows that adaptive perfusion has a good discrimination capabilities for those formulations. And we also compared the adaptive perfusion with traditional dialysis methods with using the pure drug solution and two formulations that have small and large globule size distributions. So as you can see from the left, adaptive perfusion, you can see how fast adaptive perfusion shows drug release compared with the traditional dialysis. And this the red figure also provides more direct evidence, shows that adaptive perfusion is a fast and discriminatory IVRT method for ophthalmic emotions, which is typically one type of the drug product. So now we are kind of confident that adaptive perfusion is a reproducible method, is an IVRT method that's reproducible and discriminatory. So after that, so we have one step forward, so where we implant the automation function to the adaptive perfusion method. So the figure on the left is our automated adaptive perfusion system, where we design our own programs that can achieve automatically like precondition process experiment and watch the system when the experiment complete. And we also develop an algorithm that can automatically maintain a constant volume in the retentive reservoir, which is, like I said, very critical to calculate the drug removal rate during the experiment. So this automation function dramatically reduces the human burden and allows the result to be consistent even if the system was operated by multiple persons. So the figure on the right shows that this automated adaptive perfusion system can generate similar result, compared with the manual operations. So this shows this system the result generated by this system is also trustworthy. So in addition to ophthalmic emotions, there's another category of complex drug product, which is formulations that can contain nanoparticles, like paclitaxel protein bonded injection suspensions. So the challenge is for this kind of formulation is that we are lacking the technology that can sensitively detect the rapid release process of these formulations. And we are exploring the IVRT method can help us understand the role of electrolyte in stabilization and release mechanism. So another category is in this case is Dr. Robinson lipsonic injections. So the challenge for this kind of formulation is that its delayed release mechanism is difficult to measure using traditional IVRT method, and also like a limited nanoparticle is kind of growing platform for the drug delivers, and we need better IVRT method to understand these formulations. So because of the lipsome shares many similarities with lipid nanoparticles, so Dr. Robinson lipsonic injection can be really serve as an ideal platform to study and facilitate the assessment of future lipid nanoparticle submissions. So here is a summary. So IVRT approach for complex drug products can be challenging and needs more concerted effort to improve. And a new IVRT method was developed that can improve understanding of the in vitro release behavior of complex drug products. And lastly, and we really encourage the development of more innovative and fit for purpose for IVRT methods. So here's the acknowledgement. I really want to thank my always mentor, Dr. Xiaoming Xu, for his guidance, and also all the colleagues and collaborators. So, and here is the challenger questions. So true or false? To develop a suitable IVRT method for complex product, compendial apparatus should be used. And the answer is false. So here is the second challenge question. So which is the following is not critical for developing a suitable IVRT for complex drug products? A, sensitivity, B, selectivity, C, robustness, and D, in vitro in vivo correlations. So, and the answer is D. So thank you for your attention. And this is all finish all the presentation. I will invite all the speakers and panelists to the stage, and we can start the QA section.
Thank you very much to our speakers, and we would like to invite our extra panelists to join us. Dr. Hee-Sung Chung, lead pharmacologist with OGD's Office of Bioequivalence, and Dr. Chaomin Zhu, Division Director of the Office of Pharmaceutical Quality Research within the Office of Pharmaceutical Quality. So as a reminder for the in-house, we have 2 mics. Please don't feel shy. Come up to the mics, we'll take turns. Hopefully we have people to take turns. And then we'll go to 1, 2, and then we'll go to an online question in the back and start the whole circle again. So let's start with that, and I will go down to the floor. Not on the floor, but you know what I mean. Anyone in-house? So exciting.
Thank you for the presentation. It's very informative. So I have 2 questions. The first one is for the polymer-based depot long-acting injectable. There are normally like 3 stages, right? Like Dr. Mostofa said, like the degradation, and then the diffusion and the burst release. So does the agency expect like one method or multiple methods to cover different stages of release? Because from our experience, it has been very challenging to have a method to mimic the actual release in vivo.
So the question is, in case of polymer-based formulation, the mechanism of drug release consists of different stages. The initial burst release, which is followed by diffusion facilitated release, and degradation facilitated release. So the question is whether we expect that the proposed IVRT method exactly mimic all these release mechanisms. So actually, as I mentioned, that we do not expect that the proposed IVRT method would exactly mimic the in vivo release profile or the release mechanism, but we want to see that the IVRT method, with the proposed IVRT method, the basic characteristics of the drug product, the basic release profile of the drug product is not changing. And there should be, if possible, we want to see that the mechanisms, this initial burst release, and then the more sustained release profile can be replicated. But if with the proposed method, if a sponsor can demonstrate that their proposed method can differentiate the factors which can affect the release profile, then with those justification, that proposed method can be considered acceptable. So we cannot actually dictate what it should be, the method for what it should be, but the applicant has the flexibility to come up with this method, and they have the flexibility to provide justification for the acceptability of their method. But yes, it is definitely advisable to try different conditions and try to see if it can have the relevance. The proposed IVRT method has the relevance with the in vivo mechanism. I hope you have.
Thank you very much for that answer. Anyone else? Normally, if we have a long line, we'll take turns, 1 question each, but it seems that you have the floor. Was somebody raising their hands just now? No hands. You got to come up to the mic, please. Let's go ahead with your second question in the interim.
So for those like long-acting injectables that like release over months, we have developed some like accelerated methods, like short-term method for dissolution. Does the agency also expect like a real-time or long-term method for like a comparison purpose or like a QC or release purpose?
So for QC purpose, definitely the consideration would be different, but for bioequivalence purpose, if it is as a part of the in vitro bioequivalence approach, then definitely, as I mentioned, the expectation is not that the release profile of the IVRT needs to exactly mimic the in vivo profile. As you mentioned, like in some cases, it can be as long as 1 month. So we don't want to, it is not practically feasible. We don't want to see, we don't expect to see that. But we definitely with IVRT method, the release profile would be expedited. But this expedited release profile should have relevance to the in vivo condition. And with expediting, it should not alter the basic release characteristics or the basic release mechanism of the drug product. Then, as I mentioned, that it can give us some artifact release profile, and that would be misleading for the comparability of the rate and extent. And so applicant has the flexibility to come up with their release profile, and under the given condition, we will see whether that release profile is actually adequate enough to potentially differentiate a non-BE test formulations. So it's like case-by-case basis, we have to see the total picture. Then we can see, we can assess that whether it is acceptable or not.
Thank you.
Thank you, Dr. Mustafa. And
Okay,
question at mic 2, please.
Okay, my question is regarding generics, like the Me Too products. Usually we look at the USP for procedures for release criteria. In case of a novel drug or something being developed, do you share, as the agency, do you share the typical processes that some of the leading companies are using to a small startup company that's trying to develop a product?
So I can maybe try to answer this question. I think also internally, it'd be great if we have a dissolution database-like system for these complex products, or if we have like a standard method that industry can use. But the reality is that we don't have that. So, you can start with certain methods, right? I think typically people may try USP2 or USP4 if it's, I'd say, emulsion product, for example. But we have also shared today that there's research on different methods that applicants may try using. So we welcome new approaches that you may be considering, and then you can approach us with those methods. But again, we don't have a set method for specific product. That also provides some flexibility for the generic industry to come up with different methods rather than sticking to one method, which may or may not work for your product. Thank you.
Thank you, Dr. Chong. Let's go online for a question, please.
Great. We have an online question for Dr. Xiaoming Zhu. Is IVRT demonstration done once only, and does that experiment replace PK study?
That's a very interesting question. So typically we know the IVRT, as you've seen today, we'll discuss a lot of novel methodologies. And the reason why we need this kind of novel methodologies is because of the challenges that we recognize with the existing IVRT method, as a previous question was alluded to about the USP method. So when we are looking at these challenges, especially if we desire the IVRT method to be used to support, for example, bioequivalence, demonstration of the bioequivalence, then we certainly recognize the difficulty associated with its development that's needed. So for this reason, so typically, at least for now, the IVRT for demonstrating bioequivalence can be considered, for in supporting of the study in the initial kind of filing submission. So certainly there's a kind of the other subsequent question is, can this method also be used for batch release, and then as part of the continuing to release batches? You would think, a good method that suitable for demonstrating bioequivalent would also be suitable for demonstrating quality assurance, QC. But we do recognize the challenge is still there, and we desire at some point to bridge those gaps. But for now, what we are limited discussion is looking at the method to supporting bioequivalence. So for those reasons, those as a one-time study with adequate justifications, with adequate support for validation in order to support that it can serve in lieu of, for example, a clinical endpoint or PK-based BE approach. So that's, I think, the current view on how the IVRT may be used. So hopefully that answered the question.
Thank you, Dr. Su. Mic 1, please.
I have a question for the last speaker. You have two questions. The least critical factor for the RT. So you said it's the in vitro numerical correlation is not critical. So I just wanted your comment. Why is that?
Thanks for the question. So the challenger question is saying that which is not critical for developing IVRT method for when you not critical for when you're developing IVRT method complex drug product. The reason the answer is D, in vivo correlation is not that doesn't mean that it's not critical. It's because like if we really using those to require the IVRT method to achieve the IVRT standard, so it will be really hard for us to for the applicant to develop such kind of like IVRT method. So it's not doesn't mean it's not critical. It's because the standard is very high. So we have to, besides to achieve the IVRT, we have to first to think about the sensitivity and selectivity and robustness and process for when we develop the IVRT method for the complex drug product.
Thank you, Dr. Su. Next question.
Thanks. Great talks today. Quick question. Microspheres, PLGA, just curious if you could get an update on any of these technologies for the IVRT are applicable towards that, or if there's anything that you guys did not talk about today during your presentations that might be an ongoing area of research. Thanks.
Yeah, we didn't present on the PLGA microsphere. So, but as you know, last year we had 2 approval of the generic PLGA microsphere product. So part of that, I really attribute to the GDUFA research program that a lot of research has been done at academia laboratory to kind of support. A major part is the how do we measure the release from those long-acting injectable products. So without going into detail, I would refer to you to some of the publication that FDA and collaborators has already published, especially in the microspheres domain. University of Michigan, Purdue, and Connecticut, and the multiple groups, and UT Austin, they have a lot of research that's in that area that we think can be very helpful for the generic development. So I will refer you to some of those published work already.
Thank you, Dr. Su. So for the benefit of those in-house, we do have several guidances, the cover sheets with the QR codes outside that are relevant to the talks that you're going to hear today and tomorrow. So please take a look at them when you step outside. Also, you're here. Please don't be reticent. This is the opportunity to ask your questions. And don't depend on catching them outside the door, because you may not get that opportunity. So please, this is the forum to ask your questions that will benefit everybody online and in-house. You're here. Don't say, I'm going to wait until tomorrow. Do it now. Online question, please.
We have an online question for Dr. Dongkai Zhu. Are there any ongoing efforts to standardize or validate the adaptive perfusion method across different labs or institutions?
Thanks for your questions. like I described, the adapter, this method we're just also exploring, and we have been testing many formulations like emulsions, and also plan to do some more work related to the protein bonded drug product for injectable suspension, or also another type of formulation. So when you're concerned with like validation process, like so it's still very critical to consider the robustness and to think about the reproducibility and the discriminatory for this such kind of IVRT, this kind of IVRT method. Thank you.
Yes, Dr. Zhu?
Yeah, just to add, so I hear the words in the question, standardization and cross-laboratory. So I think there are different aspects. A standard, when we're talking about standard that require, a significant effort. So for example, USP or ASTM or ISO. So those, I think at a higher level, that means the methodology, equipment, and all the associated, practices are can be replicated in other laboratories. So, and then there's also that the method is reproducible in another laboratory, which is certainly a relatively lower bar to achieve. And I think as what we are presenting today on some of those methodologies is really not to intend to show, this is a method you need to use as if now, but rather as an encouragement for you to consider developing your own methodology, maybe using this as a starting point as you are developing the methodology. And then certainly we cannot anticipate what the product you're going to use, which means what evidence may need to be provided in order to support the approval decision. So those, we think it's your responsibility to take a step further from any of the methods that's described today, or your own method that you have developed in-house, to really show that the method actually can tell the agency what the results is trustworthy, it's reproducible, and others. So I think as of now, what we are doing internally, we're continuing the research. We certainly want to make sure the method can be used by others, but it's not something that FDA are necessarily primed to do, of a develop, a standards that, we certainly want to share with you, but ultimately to adopt, to use it, it's really up to you to use it. And then again, we really encourage you to develop your own methodology if the existing methodology is not suitable. So that's something, I want to really come to comment on that. Hopefully we can see more novel methodologies available.
Thank you, Dr. Su, for that additional perspective and exhortation. Okay, mic one, please.
So it's a really great talk, and thank you very much. So my questions came from some challenge we have. Let's say we develop product, gel or depot, and use it surgically, not just like topical. And extended release, local release, critical. And in that case, in vivo release, the mechanism, the rates are very unique for the product. I can have two products, they have very different release, but developed in vitro method, they might be the same. It's my question is, what are your thoughts? How can I develop this in vitro method to reflect somehow the in vivo method, the in vivo mechanism, so I can tell these two drug products equivalent or not from in vitro study. So your thought about that?
So the question is, I would like to repeat the question first. So so the two products that are developed, maybe in vivo they are not equivalent, but by doing the in vitro study, we found that the release profiles are similar. So the challenge is how to tell that they are actually similar, right? Am I right? Okay. So that's why the in vitro BE approach is actually the totality of evidence. So here we have it is a multi-pronged approach. So it is not entirely dependent on the comparability of the rate and extent of drug release, but also there are other components of it, which is in this case, formulation similarity, the Q1, Q2, the qualitative and quantitative sameness of the formulation. And also another important aspect is the physicochemical characteristics. So if you see the PSGs of this drug product, there are multiple physicochemical characteristics that we want to see that they compare between the test and reference product. And with addition to that, we will also want to see that with IVRT, and definitely that IVRT should be adequate enough, and we want to see that it has the discriminatory ability to detect a potentially non-bioequivalent test formulations. So based on all these different approaches, we can mitigate the potentially major bioequivalence failure moods. There are different, there are multiple ways that we can actually make a formulation which is not bioequivalent. And by cross-checking all these different things, we are actually kind of mitigating the risk of approving a non-bioequivalent formulations. So that's why it is a totality of evidence approach. And in addition to IVRT, multiple factors are considered here, multiple things are checking out. And for IVRT method also, since we want to see that under the given conditions, the IVRT method, is it actually really adequate enough to identify any potential non-bioequivalent test formulations? And that's why IVRT method validation is very critical. So in many cases, we found that the deficiencies are related to IVRT method validation. So the IVRT method validation, we found that the critical factors, either formulation related or manufacturing process related factors that can affect this depot formation and drug release were not considered in the IVRT method validation. Since the IVRT method is not validated adequately, we actually usually ask applicant to come back with the supporting evidence that the IVRT method is validated. So that's how actually we want to make sure. But definitely the best, the most confirmatory thing to do would be the in vivo B study. But by giving, and definitely when we are actually giving the options of in vitro B approach in the PSG for any given drug product, we are actually, before doing that, we are trying to make sure that whether by taking this in vitro B approach, is it practically feasible to ensure the bioequivalence of the drug product. So that's why for not all the drug product, we are recommending in vitro B approach.
Thank you very much, Dr. Mustafa. Okay, mic 2, please.
I'd like to add actually.
Oh, I'm sorry.
I'm not sure if your product is actually like same dosage form or intend to be different dosage form, but want to show bioequivalence. Then I think, whether you're going for ANDA pathway or 505(b)(2) pathway, I think there's a difference, right? So if you're thinking about a generic product, then the starting point is like Dr. Mustafa mentioned, you want to start with the Q1, Q2 product. In case of topical product, then we start with the no difference standard, right? If you go back and listen to CRCG presentation by Dr. Sam Rainey from ORS, he gives a very good presentation about how to do the IVRT study, what to consider in developing the method, etc. And so he would then, he goes into explaining you could have different formulations that come back and show same results in IVRT, but that's not what we are looking for in the generic area. And so for generic field, you want to start with the Q1, Q2 formulation for these complex products as applicable. And oftentimes PSG will indicate whether you should start with the Q1, Q2 product, right? And then use that and then develop the IVRT method, and at the same time ensure that your IVRT method is discriminating the difference in formulation, different strength, different manufacturing processes, so then the method is sensitive to detect the difference, and also then at the same time confirm that test product and the reference product are bioequivalent to each other. Thank you.
Thank you, Dr. Chang. Okay, we have time for one last question from Mike 2, please.
Yeah, thank you for the nice presentation and discussion. So I have a technical question about the same condition to study the release rate from complex formulations using these advanced IVRT methods that you described. Most of the water-soluble drugs, the driving force, mainly concentration gradients from this complex formulation. But for hydrophobic drugs, maybe this formulation is distributed in the body as is, and the release mechanism in the body might be depending on biological factor. So maintaining a same condition during, for example, adaptive perfusion or tangential flow filtration or electrophoresis may have a detrimental or destructive effect of the formulation. So how we can develop a method that is give us like a release rate that can be used for quality purpose. However, it has no relevance to the bioperformance.
Yeah, so it's a great question. I really love the topic about sync versus non-sync conditions. I think it's a great debate. We know typically for solid oral drug product, sync condition is very critical because maintaining the GI, the drug have enough solubility, dissolution rate before it can be absorbed. However, we know for some of the complex product, particularly delivered through very complex route of delivery, such as through topical, through ophthalmic, inhalation, locally acting, and the list can go on, that the limitation of the exactly following the same conditions may actually impose a constraint to the drug release that may or may not be relevant to the actual condition of use. So I'll give one example. For the ophthalmic, let's say you deliver a suspension product, and the suspension, let's say, is about submicron in the nanometer size range. And then you have another formulation that is 5 micron in size. Now, if you run a sync condition experiment of a dissolution using apparatus 2, for example, you will see equivalent release profile in that measurement condition. Now, the question is that results meaningful or useful? If you know the reason you develop a nanoparticle formulation is to enhance the rate of dissolution, because the suspension you apply to the eye only has about less than 2 minutes of time to stay on the eye surface. So ensuring that the drug can dissolve very rapidly in that short duration is more important than to measure the complete release, let's say within 1 hour of a dissolution testing, right? So this example shows that maintaining the same condition, let's say, to ensure you testing 2 products, if you actually get the same results, is that meaningful? So in this case, non-sync condition may actually tells you what is a tendency for the drug to dissolve in a very rapid fashion. Actually, we have published some work related to this that, even a few hundred nanometer differences, you can actually drive the drug release to see the difference. Some can dissolve within 10 seconds, which is very desired, right? And some will release in 5 minutes. So then, relative to the eye condition, the residence time on the eye surface, when you put all of this information into consideration, you need to make a determination which is more important, which is more relevant. So certainly, certain conditions, sync condition allow you to have more reproducible results, but also at the cost may not be relevant or provide useful information. So it's something that really need to be considered during the development. And as part of your submission to the agency, don't be afraid to put in the justification. Don't be afraid to put in justification and reasoning to say, why this is not needed and why this is useful. So it's really case by case. So, I didn't answer really your question, but it's this topic that I think need more discussion, needs more, I think, consensus about, what is really important.
Okay. Thank you very much. Thank you to our panelists. This brings us up to a break.